Hello everyone, my name is Gary O'Mealy, and in this video we are going to wrap up our discussion on macromolecules with a video on nucleic acids. So nucleic acids, which of course encompass both DNA and RNA, these are polymers that are constructed by attaching nucleotide monomers together by dehydration synthesis. Nucleotides themselves have three basic components. First, they consist of a pentose monosaccharide, deoxyribose in DNA, or ribose in RNA. You'll notice that the difference between deoxyribose and ribose is that deoxyribose is missing its 2' hydroxyl group. If the 1', 2', 3' numbering system of sugars like this is new to you, the carbon that I'm pointing at right here on the right side of this oxygen atom here is the 1' carbon, and then as you count around the molecule, 2', 3', 4', and then 5' right here. So deoxyribose is missing its 2' hydroxyl group, so if we look at ribose, you can see that 1', 2', there is a 2' hydroxyl group here, whereas that hydroxyl is replaced with a single hydrogen atom there, and that is why deoxyribose has its name. Next, every nucleotide is going to have a phosphate functional group attached at the 5' hydroxyl region of the sugar, and this phosphate, carrying strong negative electrical charges, is going to make sure that every nucleotide and every nucleic acid is strongly negatively charged. And then finally, every nucleotide is going to have one of 5 nitrogenous bases, which we will introduce in the next slide. The base is attached to the 1' carbon of the sugar, and the identity of the base helps to contribute to the information that is coded into DNA and RNA. So here we can see the 5 different nitrogen-containing bases. They are classified as either being pyrimidines, which are cytosine, thymine, and uracil, or the purines, which are adenine and guanine. So the important thing to understand here is that these bases contain polar functional groups that are capable of forming hydrogen bonds, and that forms the basis of what we see when we see complementary base pairing, especially between 2 opposing strands of DNA. So when we're actually building a nucleic acid polymer, we need to do dehydration synthesis between 2 nucleotides to get the process going. The chemical bond that we are going to form with dehydration synthesis is going to occur between the 3' hydroxyl of the sugar and the 5' phosphate of a different nucleotide. So you can see here that the attachment point is going to be, it's not properly illustrated here from your textbook, but if we back up for just a second, here we go. So when we say the 3' hydroxyl of one nucleotide, we're talking about this hydroxyl right here in ribose, or this hydroxyl right here in deoxyribose. So the bond we're going to form is between the 3' hydroxyl of one sugar for one nucleotide and then the 5' phosphate of a different nucleotide. So when we start to polymerize our nucleic acid, the bonds that we are forming are going to alternate phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, with this end of the strand where the phosphate is exposed being the 5' end of the strand, and this end of the strand with the sugar exposed being the 3' strand. The bonds that we are forming are called phosphodiester bonds, and the entire backbone of the strand is often called the phosphodiester backbone, and you'll notice that the nitrogenous bases are just kind of hanging off to the side here. They do not participate in the formation of these phosphodiester bonds, and this leaves them free to participate in complementary base pairing via hydrogen bonding with one another. And as we saw with protein synthesis is unidirectional, always from N-terminus to C-terminus. Nucleic acid synthesis is very much the same way. It is unidirectional, always proceeding 5' to 3'. Moving on to complementary base pairing, you can see this fairly well illustrated here. Since the bases do not participate in the phosphodiester backbone, they are free to form hydrogen bonds with one another. However, the bases cannot hydrogen bond with one another in any combination. There are rules that we have to follow, and these are often called Chargaff's rules. Adenine, which is a purine, will always hydrogen bond or base pair with either thymine, which is found in DNA, or uracil, which is found in RNA. And guanine, another purine, will always hydrogen bond with the other pyrimidine, that being cysteine. So you cannot get A pairing with G, you cannot get C pairing with T. Another thing you'll notice here is that the base pair between adenine and thymine consists of 2 hydrogen bonds, whereas the base pair between cytosine and guanine consists of 3 hydrogen bonds. This does indeed mean that the base pair between cytosine and guanine is about 50% stronger than that between adenine and thymine. Finally, let's wrap up by talking about some of the differences between DNA and RNA. They are both built in very much the same way by attaching nucleotides together, forming phosphodiester backbones by dehydration synthesis, but we'll outline some of the major differences between DNA and RNA here. Human DNA contains the roughly 30,000 different genes worth of genetic information on how to build all of our proteins, and this information is spread out over 23 unique chromosomes for which we have 2 copies of each, one from mom and one from dad. RNA, on the other hand, is used more as an intermediary in the process of synthesizing proteins from those genes. That's how we get things like messenger RNA, transfer RNA, ribosomal RNA. Those are all just kind of intermediary tools that we use to build the proteins from a DNA-based blueprint. So this chart here will lay out some of the major differences between DNA and RNA. DNA, of course, is double-stranded, whereas RNA is usually going to be single-stranded. DNA, we already saw, uses the sugar deoxyribose, whereas RNA uses ribose instead. In terms of which nitrogenous bases they use, DNA nucleotides are either A, T, C, or G, whereas RNA nucleotides are either A, U, C, or G. DNA uses thymine, whereas RNA replaces thymine with uracil. And as far as the length of the polymer we can expect, DNA molecules tend to be very, very, very long, at least 1 million base pairs, sometimes close to 1 billion, whereas RNA polymers tend to be much shorter, usually hundreds, usually not much more than thousands of nucleotides at a time. So as far as complementary base pairing goes, DNA strands base pair with one another, and that is what leads DNA to be double-stranded, whereas RNA strands tend to base pair with unwound DNA like what we see in the transcription process, or other RNA molecules like what we see in translation when we see temporary complementary base pairing between messenger RNA and transfer RNA, which we will be talking about in future videos. And then since we're on the topic of nucleotides and nucleic acids, more so nucleotides than nucleic acids, let's talk about some high-energy nucleotides. The standard nucleotides we've been talking about that are used for DNA and RNA synthesis have only one phosphate that gets incorporated into the phosphodiester backbone. This phosphate is stably attached to the 5' hydroxyl of the sugar portion of the nucleotide. High-energy nucleotides like ATP and GTP have extra phosphates attached here. They don't just have one phosphate, they have up to 3 of them. If we look at the chemical structure of ATP, you can see all the usual components of a nucleotide, including the adenine purine base, the ribose sugar, and then the phosphate, but again, we see 2 extra phosphates listed here, and you can see the extra chemical bonds that are involved here, which are highlighted in red. These are very unstable and therefore very high energy bonds. These 2 extra phosphates are known as the gamma phosphate on the end and the beta phosphate in the middle. The alpha phosphate, which is right here, is very stable, and you're not usually ever going to remove that. Each red-line chemical bond, as I said, contains a vast amount of energy that, if broken by hydrolysis, will release energy into the system that can potentially be put to work to get other things to happen. The hydrolysis of ATP into adenosine diphosphate to release one of those attached phosphates has a delta G value of negative 31 kilojoules per mole. This is an exergonic reaction, very highly spontaneous, and this will release 31 kilojoules of energy into the system. Again, this energy can be potentially used, funneled into some other kind of reaction to pay the energy cost to get something to happen. This is why it's so important that ATP is produced inside of active cells. Not every physiological process is going to be spontaneous. Some processes like muscle contraction and membrane transport require energy to occur because they are not favorable reactions or not favorable processes. So the cell needs to have ATP on hand to basically act as currency to get these things to occur. So hydrolysis of ATP into the lower energy ADP form or the even lower energy AMP form will release energy that can be funneled into anabolic or endergonic processes that require some sort of energy payment. These are called ATP-coupled reactions. If we imagine an anabolic reaction that takes the form of A plus P to give us AB, and if we say this reaction all by itself has a delta G of positive 25 kilojoules per mole, which is non-spontaneous and endergonic, if we couple these two reactions together, meaning that they happen at the same time and in the same vicinity as one another, the overall delta G for this coupled reaction is still exergonic and has a negative delta G value. So in doing this, the hydrolysis of ATP pays the energy cost directly for the anabolic endergonic process to occur. This again will include unfavorable but necessary physiological processes like muscle contraction, membrane transport, and things of that nature. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, compare and contrast DNA and RNA, both in structure and function. What are the three components of a nucleotide and which ones are actually participating in the formation of phosphodiester bonds? And lastly, how is ATP used to pay the energy cost of anabolic reactions? So that's going to do it for this video. If you have any questions, leave them in the comments section. If not, I will see you next time.